Hello, Dr. Neeraj.
Hello, ma'am. How are you?
Good morning, ma'am.
Good morning. Good morning. I hope you circulated it to all your students.
Yes, too many.
And also to the, yeah, thank you. Thank you.
So we are live, ma'am, now.
Okay. Okay, good morning, everyone. And good evening to Dr. Neeraj, Dr. Neeraj Sharma, who is assistant professor at the Genetics Institute in Johns Hopkins University. His work has mainly concentrated on genetics of cystic fibrosis. And we are very proud to say that he completed his PhD from PGI Chandigarh. And then he went on to work in Dr. Gary Cutting's lab at Johns Hopkins University, where he's done a lot of original work and has been associated with a project called CFTR2. And we have Neeraj with us today. And what we are going to do is to have a module on this disease, cystic fibrosis, in which we will have a series of lectures. Dr. Neeraj, please.
So I'm going to share my screen here. Can you see my screen?
Yes.
So good morning, everyone. And let me have this in a slideshow mode. All right. So good morning, everyone. It gives me an immense pleasure to talk about this disease, cystic fibrosis. And one of the reasons for this is that my journey in this disease, cystic fibrosis, started at PGI. As you know, that during my PhD in the Department of Biochemistry, for some reason, yeah. So what I was doing at that time was measuring sweat chloride in individuals who were suspected to have signs and symptoms of this disease named cystic fibrosis, and I would measure sweat chloride in those individuals. During my PhD in the Department of Biochemistry, what I found that CF existed in India. There was a different spectrum of mutations in CF as compared to what we see among the Caucasian population. Frequency of delta F508, however, which is one of the most common mutation among Caucasians, in Indian population, we found that it was only 20 to 30%, which suggested that the spectrum of mutations in Indian population was quite heterogeneous. We also published a review, Epidemiology and Genetics of CF in Asian countries, emphasizing on the role of how we should move ahead with functional characterization of these variants among Asian populations so that they can be prioritized for the precision medicine. So I'm going to give you a brief background on cystic fibrosis in the context of just the words here, which are highlighted.
Your slides are not moving. I wonder if
they're not.
They're supposed to move. Yeah. We are on the first slide.
Oh, really? I don't know. In mine here, they are. So
are they on the slideshow mode? Please
yeah,
put them on the slideshow mode.
Yes.
They are in slideshow mode.
But actually, they are not. Here they're showing in non-slideshow mode. It's the shortest view.
So let me
just upload it again. It will be fine.
Yeah. Can you see this right now?
Yes. Yes, this is the first slide, but not in slideshow mode again.
Yeah. So what about now? Is it in slideshow mode right now? Can you see it?
No, it's in the same sort of mode. What you can also do is maybe select each slide and then project it, because Somehow the slideshow mode is not coming here.
But it's going on at my end. I can see it. It's going in the slide mode. I don't know.
If it's not a very heavy presentation, you could send it to by mail and we can upload it from here, or you can just select your slide and maybe go back to the slide sorter view and select your slide and then click on it.
I'm
It'll be
doing
done.
it. Yeah. I am on a slideshow mode right now. Now, can you see it right now? I'm on a slideshow mode right now.
Yeah, but somehow it's not coming here in the slideshow mode. And probably that's why they're not moving.
Dr. Neeraj, I would suggest you close this presentation and then open it again.
Could be, yeah. That would be.
Thank you, Dr. Indu, for coming.
Thank you. It was really, I was waiting to attend this presentation.
Oh, thank you so much.
Pleasure. Let me do this first. I'm sharing the screen again.
Yeah, you're screen sharing, but again, not in slideshow. You click on that slideshow thing.
Yeah,
below.
now I'm.
Yes, now it is.
Now it looks good.
Yes, yes. I think
Okay.
we're moving now. Yeah.
Okay. Okay, so I start again. Sorry
Yes,
for that.
I think we should record now. Sorry. Yes.
All right. So I'm going to start with from where my research in this field of cystic fibrosis started. And that is at PGMIR Chandigarh, where I did my PhD in the Department of Biochemistry, where what I was using at that time was one of the methods for the diagnosis of CF, and that is sweat testing. So the individuals who were suspected to have signs and symptoms of this disease, cystic fibrosis, they were sent to the Department of Biochemistry by Dr. Meenu and her colleagues in the Department of Pediatrics. We would measure sweat chloride in these individuals. And since sweat chloride is a diagnostic marker for the diagnosis of CF, we would prioritize those patients who would have elevated sweat chloride and to perform CFTR sequencing on those individuals. What we found and emphasized on this point that CF existed in India, because it is primarily considered as the disease among Caucasian population. What we also observed that the spectrum of the mutations in cases of CF and individuals with congenital bilateral absence of vas deferens, which is one of the phenotypes among CF individuals, they had a spectrum of mutation that was quite heterogeneous. Delta F508, which is one of the most common disease-causing variants among Caucasians that occurs at a frequency of 90%, of which 50% are homozygous. Among Indian population, the frequency was quite low, around 20 to 30%. What we emphasized in one of the reviews that we published in the Journal of Respirology with Dr. Meenu was that there is a need to functionally characterize all the mutations that you identify among Asian populations or low middle income countries so that they can reap the benefits of precision medicine. I will come back to this point later. So giving you a brief background on cystic fibrosis, just in the term of words here, what CF is, it is an autosomal recessive disorder, which means it is a genetic disease, which is inherited from the parents. Parents are carrier, and they lead their normal life. However, the unfortunate children that gets one copy from each parent has to deal with this devastating disease, cystic fibrosis. The mutations in the CFTR gene are responsible for dysfunctional CFTR. CFTR protein otherwise is a chloride channel, but when it gets dysfunctional, it results in a multi-system disorder. What I mean by multi-system disorder can be seen here in this particular image, that it is not only one organ like lung, it also affects pancreas, liver, reproductive organ, sweat gland, and other upper respiratory tract as well, and even in the intestine. The diagnosis of CF is elevation of the sweat chloride. It could be greater than 60 milliequivalent per liter, but there are certain variants that can manifest with low sweat chloride as well. But then there is identification of two disease-causing variants. When this comes in a combo, that gives you a typical case of cystic fibrosis. These are just the words. I know it becomes a little difficult when you put it in the context, but just imagine that in 1989, when this disease-causing gene was identified, it was quite an uphill task because, as we know, the genome is quite big. So these three people, they collaborated together, which emphasized the importance of how collaborations in science work. On the left is Lapchi Choi, who is a geneticist, at that time working at Hospital for Sick Children in Toronto. In middle is Jack Ruiden, who is a structural biologist. And on the far right is Francis Collins, who is a medical geneticist, at that time working at University of Michigan, just last month retired as director of NIH, and now reappointed as science advisor to the President of the United States. Just showing here all three males, but if you look at those journal Science where 3 consecutive papers had come, the first authors are women, Bachiva Kiram and Joanna Romans. It is very important for us as a scientific community to respect the diversity and inclusion in science, and women play a great role in that. How Francis Collins described his pioneer discovery was like finding a needle in the haystack. And one could imagine that there are 20,000 genes and vast introns and other sequences to find one causative gene responsible for this devastating disease was just like finding this needle in the haystack. So now, because I said like that cystic fibrosis, I put that in word. Here I will try to show you an animation if it works. I will give you a brief tour of moving from the pharynx into the trachea to the bronchi, and then I'll zoom this to give you a better stylized fashion of how CFTR works. So here we are moving from the pharynx into the trachea, and then the bifurcation of the bronchi. We are going to the left bronchus here, and here is a magnification of the bronchus here. And here you see the epithelial layer on top of which cilia are beating, surface liquid is moving. And if I give you a stylized version of a CFTR protein, if you will, what this is showing in yellow is that the chloride ions are moving out. When chloride ions move out, it keeps the airway surface liquid well hydrated, because of which the airways are fine and healthy. What happens in case of CF, that this CFTR protein becomes dysfunctional. As you can see that these yellow balls are no longer moving. What that results in case of a diseased state is the cilia no longer moves because the secretions become thick and viscid. Chloride is not moving out, no water is there. And when all this situation happens, it results in an inflamed state because colonization of the bacteria happens. And that is one of the hallmarks of CF disease. So as I said, delta F508 is one mutation. What has happened over the time is that more than 2,000 variants in CF have been reported. At Hopkins, we are maintaining a CFTR mutation database with functional characterization, and we call it as CFTR2. What we are showing here is the distribution of these CFTR variants that fall into various categories. There could be an RNA processing defects, a missense variants, there are certain synonymous variants, and there are complex variants as well. Among RNA processing, you can see here that there could be a splice variants, out of which some are canonical splice, which are easy to predict. There could be deletions, duplication, variants in the promoter region. There are nonsense mutations and frame shifts. Majority of my talk today will focus on these RNA processing defects for the reasons that I'll come to my next slides here. So in this, stay with me because I'll walk you through this slide, which will become a little bit more complicated, but I will come back to these concepts later as well. So that will be easy for you to understand. What I'm trying to show here is that how our understanding of the molecular genetics of CFTR has resulted in the improvement of the median predicted survival of individuals with CF. And this concept could be called as theratyping. So here, what I'm showing is that these CFTR mutations could be divided into different classes. The first class here is class 1, in which when DNA is transcribed into mRNA, in class 1 variants, because of these PTC mutations, this mRNA is not stable, and it results in a nonsense mediated mRNA decay, and that is called as class 1. There is also a class of variants that will have some effect on the expression of the mRNA, because of which less amount of the protein is going to be synthesized, and we bin that as class 5. And the examples for this could be a nonsense mutation, for example, W1282X. An example for class 5 is an intronic variant, and this reminds me of one girl at PGI, which we had seen, and Dr. Mean, you might remember, she was like an 18-year-old in which we had found this particular variant and had presented this case to Jeffrey Kurland. At that time, we did not know what the other variant in her CF gene was. But the concept here is that this particular variant belongs to class 5 because of the splicing defect and because missplicing is not complete. Moving next to the class 2 mutation, what happens in this case is that there is no defect in the mRNA processing. However, the variant in this class results in misfolding. Because this is misfolded protein, it never makes to the Golgi, which is shown here in this pink organelle. So because this protein is misfolded, this is sent to proteasome for degradation. And that is class 2. And the classic example of that is F508del, which is the most common disease-causing variant among CF population. And then there are classes 3, 4, and 6, where the protein goes to the surface. It makes it to the surface of an epithelial cell. However, there are either the gating defects, which means that the chloride is not gated enough to go out to the apical side, or there is a conduction defect, that it is not able to conduct properly. And in class 6 mutation, what you see is that there is a rapid turnover of the CFTR protein. In case of normal functioning, you see that the chloride ions can move. So nutshell here is that in case of a dysfunctional CFTR protein, enough chloride ions do not go out. And the examples for these are listed here. So these are all classes of the mutation. How our understanding of this that we have bended has helped in in increased survival of the patients with CF is, it's not moving further. Yeah. So this is one class of the drug that has got FDA approved, and this is called as potentiator, that works on the variants like class 4, 3, and class 6 that make it to the surface, and it results in the potentiation of the CFTR protein. More recently, the drug that has come up that has been used to treat F508del, and that covers now almost 90% of the patients with CF, is a combination of the drugs in which it corrects the misfolding defect, as well as it potentiates the CFTR protein. So it is a combo of these drugs that is used to correct the defect of F508del. What this has resulted in, as you can see, like the time point when the CF gene was identified to the current times here, how the median predicted survival has increased. What is even more fascinating is that look at this time point. Yesterday was the 10th anniversary for the first use of this modulator drug potentiator, how this graph has increased tremendously in the improvement of the life of these individuals with CF. Important point to note here is that these modulator drugs, they only work for those variants that result in the production of the CFTR protein. Unfortunately, individuals who belong to this class 1 variants that do not result in the production of the protein because it undergoes nonsense mediated mRNA decay, those individuals are not prescribed these variants. So what we should do for that? So what I have done here and at Johns Hopkins is I have undertaken a systematic evaluation of these premature termination codon variants to inform therapy. And the questions that I have asked and I continue to ask are that which of these PTC variants make stable mRNA? And which PTC variants among those can make some mature truncated protein? What is their level of CFTR function? Whether they can be activated or corrected by FDA-approved CFTR modulators, whether NMD inhibition can augment CFTR modulator response, can we use base editing approaches for these PTC variants? I'll try to give data on some of these questions that I have been asking. How do we study these PTC variants? Many of you here in the audience, they may be studying their molecular biology by expressing their gene of interest in the way that they have an open reading frame and they express the cell in the cells and ask the questions, what kind of a protein is made? But to study these nonsense mutations, frame shift variants or splice site variants, the central paradigm for that is that it has to undergo splicing. And for splicing, we have to have introns introduced into a cDNA context. So here is an illustration of a CFTR gene with black boxes showing in exons, and the single line here is reflecting the intron. What I have done is that I have introduced either full length or abridged introns into the context of the cDNA. I call them as expression mini genes, and I have made them in a different iterations. So you can call this as expression mini gene intron 1 to intron 5, then 10 to 12, 14 to 18, because all of the introns cannot be introduced. In these expression mini genes, then I went ahead and then I site-directed mutagenized them to create nonsense variants, frame shift variants shown here in red or purple, intronic variants shown here in blue, certain missense variants that are predicted to result in splicing defects. So in total, I have so far studied 26 nonsense mutations and frame shift mutations. 15 exonic and 35 intronic splice site variants. And all these have been shown in different publications in last 7 or 8 years. All right, so that is the expression many gene concept. But what kind of a model systems, like cell-based model systems, you would use to assess their first disease-causing potential and then to test the modulator responses? So one is some cell line model systems where we heterologously express these variants. And here we have used a flip recombination-based method where a CFTR expression mini-gene is co-transfected with the recombinase into a cell line, which is called as a flip-in host cell line. And it is zeosin resistance is there in the flip-in host cell line, but when it gets co-transfected and recombination happens, what it results in is the loss of zeosin resistance and gain of hygromycin resistance. And that results in the stable cells that are expressing CFTR variants. And then you can test CFTR modulators, read-through drugs, NMD inhibitors. You can try gene editing approaches and also splice modulators. The outcome variables here could be RNA that you could do by simple conventional RT-PCRs. You can do bulk RNA sequencing. You can study protein by Western blot and measure chloride channel function. The second model system that I have used is nasal epithelial cells, which is very important from individualized studies point of view. What I do here is that I collect the nasal cells from individuals with CF and their parents. Nasal brushes are harvested, they are co-cultured. And here you can see that these HNEs, they come up as islands of cells on a mouse feeder irradiated cells, which are 3T3 cells. And these islands of cells are reflective of human nasal epithelial cells that grow, and these are observed under phase microscope. Once you achieve these islands of cells, they are put on airway liquid interface culture so that it mimics the in vivo situation, as you see in the nose, that there is air, and then there is a basement membrane that through which all the nutrients are fed. They are allowed to differentiate. And what we mean by that differentiation is, you look here, there is appearance of cilia. This is scanning electron microscopy on one of the filters from one of the individuals with CF. Look here, the cilia that comes up, and this shows that the cells have got differentiated. And at this time point, then we measure either CFTR function, we perform RNA studies, look for splice isoforms, and do immunoblotting to look for CFTR processing. In terms of chloride channel function, how do we measure those? So these cells that are grown on the filters, they are mounted on an instrument called as Using chamber. So these are two chambers here. And in this slider, these filters are inserted. And you can see here is a cartoon of how the cells are arranged on these filters. And this side is apical, and this side is basal, and these arrows reflect the movement of the ions when these chambers are filled up with a buffer. It could be a gradient buffer, or it could be an isotonic buffer here. So then how do we measure the function here? So here I'm going to give you a cartoon. So on x-axis here is the time, and on y-axis is the short circuit current measurements. When the voltage is clamped on Using chamber, the flow of ions is allowed to equilibrate, shown here in this stable line here. Earlier it is messy, and then it gets stable over the time. And then we add amiloride. Amiloride is an inhibitor of epithelial sodium channel. Once that ENaC activity is inhibited and this short circuit tracing again gets stabilized, now the trick comes where we add forskolin and IBMX that increases the cyclic AMP levels. And we know CFTR is a cyclic AMP mediated chloride channel. When cyclic AMP concentration increases, cyclic CFTR activity increases, and that is shown here with this line here. And then we add inhibitor 172, which is CFTR specific, and then you see that the short circuit tracing comes back to the baseline, and then we measure the difference between these two, and that is reflective of CFTR function. We call it as delta ISC. So this is in case of a non-CF, nasal or bronchial cells. On the right, I'm showing you how a CF cells look like. And as you can see here, that on adding forskolin and IBMX, there's virtually no response. And it is almost a flat line, and delta ISC is like a few to no micron of CFTR currents here. So now I'm going to give you a kind of another animation to give a perspective of what I showed you in the last slide here. So here is the CFTR molecule on the surface, and this is this is a dysfunctional CFTR. There is no movement of the chloride ions onto the apical side, and that is what we measure, the transfer of the chloride ions across this epithelial cells. So in CF individuals, there is no movement of the chloride ions. So now small molecule correctors, how do they correct the folding defect and they bring more CFTR protein to the surface? This is a 2D representation of that. Here you can see that only few or no CFTR molecules are present on the surface. But when you add those corrector molecules that I was showing you earlier, it could be lumacaftor, tezacaftor, alexacaftor, but these are in combination. If you use only one, it doesn't work. What that results in glycosylation of that CFTR protein so that it gets fully glycosylated. It goes through the Golgi, reaches the surface, and it starts conducting the chloride ions. Again, putting this in the perspective of an animation here, as you can see that what happens with the delta F508 or core glycosylated protein, it gets ubiquitinated, never goes to the surface. However, when you add the corrector molecules, it allows it to fold, reaches the surface, and now you start to see the movement of the chloride ions. This looks good in animation, 2D concepts, making the illustrations, but how does it look like when you do a real lab experiments here? So here I'm giving you an example of a stable cell line that was created and that expresses this F508 del. On the far left here is that F508 del, which is untreated. And on the far right are the controls. The wild type CFTR, as you can see here, just focus on the two bands here. The very strong band here on the right with the wild type CFTR is the complex glycosylated CFTR that goes to the surface. And this is that very strong band. It has more sugars, that is why it has a higher molecular weight. It also makes a core glycosylated protein which runs at a lower molecular weight of around 143 kilodaltons. Interestingly, what you see with F508 del and what I have been trying to show in all the animations here was that it is core glycosylated. It never goes to the Golgi, no sugar molecules are added, because of which it only makes a core glycosylated protein, which is a 143 kilodalton. So on the left, the experiment that we have done, F508 del, recapitulates our control lysate. It only makes a core glycosylated protein. Interestingly, what you see here is that use of the corrector molecules, Luma, Teza, or alexacaftor, by itself, they do not give any benefit. It still remains core glycosylated, as you see with these bands. However, when a combination of lumacaftor and alexacaftor or tezacaftor and alexacaftor, when these two corrector molecules are made together, you start to see that this F508 del makes complex glycosylated CFTR protein like wild type. This is on Western blot. Now, how does it look like on a real functional study? This is on a primary human nasal epithelial cells of an individual who had F508 del on one of its allele. And it is quite remarkable here that when you do not treat these cells with the corrector molecules shown here in the blue line, after the ENaC inhibition with amiloride, there is a sharp fall in the short circuit tracing. But after that, when you add forskolin IBMX shown here in blue, there is minimal to few microamp increase in the current, which is reflective of F508 del. However, when elexacaftor and tezacaftor were added, in addition to the potentiator ivacaftor, you start to see a remarkable recovery of the CFTR function as shown here with the green line, and it goes to the about 10 microamp, stabilizes, and further gets potentiated with ivacaftor shown here in this black arrow. And with using the inhibitor I-172, which is specific to CFTR function, you see a drop fall in this short circuit tracing reflective of that these currents were specific to CFTR function. How much this translates to recovery of CFTR function? This is like around 75% of wild type CFTR. So this is remarkable. This corrects the basic defect in individuals with CF who harbor F508 del variant. So this is an example of a class 2 where there is a processing defect. I will give you an example of those variants. Please recall in that first slide, I was showing you those variants that go to the surface of the protein and they do not have any processing defect, but what they do not do is that they are unable to conduct chloride ions because of their gating defect. That's why these chloride ions are no longer moving out of this epithelial layer here. An example for that is G551D. However, when you add that potentiator ivacaftor, it opens up the gates. Chloride ions move from the intracellular to the extracellular side, and that corrects the defect. Again, this looks like in a very 2D illustration. How would this look like in an animation? Here I'm showing you that because this particular protein will not have a processing defect, it will go to the surface of its own, will reach the surface, but will not be able to conduct chloride ions. However, when you add the potentiator shown here in purple, it starts to move the chloride ions out. Again, these were the illustrations, animations. How does this look like in a lab experimental setting? So we got these individuals, these are siblings. They both had this particular genotype, and the first variant listed here, Q359R, belongs to that category that this has some gating defect. And what I'm showing you here, I will not go through all these traces, but what was remarkable for us was that both siblings showed a dramatic response to the ivacaftor shown here in the purple line. You can see both on the top and the bottom panel, this purple line far exceeds in terms of the height, as you will, when ivacaftor was added, which shows that the CFTR function was recovered. How much this correlated with recovery of the CFTR function? It was 30%. What was very interesting and reassuring to us, especially me, was that these individuals were not included in those list of variants for which this drug could be given. But our physicians here were able to convince the insurance, and both the siblings were put on drugs. And this is the note that we got from the mother of these individuals, that the last round of labs and PFTs came back with astonishing results. Both boys blew over 100%. Their sweat test came back to 0. And this is quite fascinating for them, and as much fascinating and encouraging for us as well who work in the lab. So this was all about those variants that make protein. But as I said, the focus of our research is moving towards those variants that do not make protein, or to select those PTC variants that can make protein. So here in this illustration, I will walk you through. It will get a little complicated, but it is easy to understand. So what I'm showing here is that it is a ribosome here, and mRNA is getting transcribed. The transcribed mRNA is getting translated here, and I have written here 2 UGAs, which means that there is a premature termination codon here. And what this would result in, if it has to follow the biological rules of the nonsense mediated mRNA decay, that the mRNA will be unstable, there will be no mRNA and no protein. And this mRNA will be degraded. So what are the options available for such PTC variants? They have to have some kind of a gene editing. Now I'm going to take you towards the N-terminus of the CFTR. And I'm writing here that these particular variants at the very N-terminus of the CFTR, they make stable mRNA. And if they make the stable mRNA, what it results is a shortened protein. This is quite remarkable, that the reason why they make a stable mRNA is because there is a downstream methionine that gets activated, and that results in the shortened protein. The N-terminal half does not get used. So that is the result of this shortened protein. However, this protein is immature, just like F508del goes to proteasome and gets degraded. However, if you use the read-through drugs, and this is an ideal target for the read-through drug that introduces a near cognate tRNA and an amino acid there that results in a missense variant, that full-length protein is formed. But because it might result in some kind of a misfolding, so they are modulator eligible. So that is the story of the N-terminal nonsense variants. There are also variants in the last exon or the first 50 nucleotides of the penultimate exon that also show features of no nonsense mediated mRNA decay, because that is the rule of the nonsense mediated decay. And the variants like E1418X, which is in the last exon, it results in a truncated mature protein, and these are modulator eligible. But biochemistry of CFTR has also shown that there are variants little up in the CFTR protein structure, like W1282X before the NBD2 domain. And we have shown this, that they result in a truncated mature protein. They are modulator responsive. However, they result in a nonsense mediated mRNA decay. So if you can inhibit the NMD, they can be prioritized for these modulator drugs. Final, in this category, there are also nonsense mutations that you should remember, and you should know that there are certain nonsense variants that result in exon skipping. This is a classic example that this sits in the middle of the CFTR, but what this results in, that either the full exon is skipped in frame, or there is one amino acid at the start of the exon that gets deleted. What that results in is a mature isoform, and that is modulator responsive. So some of these, like the ones in the last exon or this splice variant, they do not need any NMD inhibition. They do not need read-through. They all are modulator responsive. However, we have to take care of, I have binned them into these 5 different classes. Imagine like the mutations, we had called them from 1 to 6. Here, just from that first class of mutations, when you study their biochemistry, we have subtyped them into 5 different classes. And we have shown this in the PLOS genetics paper, and we have given all the data. I'll only show 2 examples, illustrative examples from this busy slide. And that is the N-terminus of the CFTR, where the read-through results in the misfolded protein, but modulators correct the misfolding defect. Here is an illustration of the CFTR gene. L88X lies on the exon 3, and now we have a data I'm not showing here in the interest of time, that it is the methionine 265 that is an alternative start that results in stable mRNA because there is a downstream methionine. And when you use the read-through drug that introduces a near cognate tRNA, in this illustration, however, it says full-length protein, but as I'm trying to convey here, that there is an amino acid that is inserted, and it is not the same leucine that will be inserted, it's a different amino acid. So it creates a missense change. So protein can be full length, but still not properly folded. And this is shown here in this Western blot quite nicely that we have done here. So on the left here, I'm showing just a control of wild type on different loadings in terms of the how much amount of the protein we had loaded. And on the very right, I'm giving you an example of F508del, that when it is untreated, it only shows a core glycosylated protein. But when you add the correctors, it results in the glycosylation of the protein. And I talked about that in the previous slides. However, this is a very interesting result here. And that is what I was trying to show, that L88X would result in a downstream methionine that gets activated, and that results in this very strong band that you see of a shortened protein. Otherwise, L88X would result in a 3 or 5 or something like that kilodalton of the protein, but this one is like close to 130 kilodalton, which shows that this protein starts somewhere from here and all these. But what is interesting here are 2 points I'm trying to convey here in this small amount of time, that when these are untreated, you see this protein being made. When you add these correctors, Luma, Tisa, Alexa, or combination, virtually nothing happens. What is interesting is when you use the read-through drug G418 or ELOX2, you start to see a protein band that comes up here, shown here as purple dot here. I'm trying to highlight this. So this becomes a core glycosylated full-length protein. This is not complex glycosylated. But when you give a combination of those corrector molecules, Alexa and Tisa, you start to see this band that starts to show up, which means that this core glycosylated protein starts to become a complex, just like F508del. So here, I know it is a bit difficult to digest in this small amount of time, but what I'm trying to show that it is the read-through drug in combination with corrector molecules that this PTC variant can benefit. So this is a Western blot. How would this look like in a functional study? So here, as I said, that compared to DMSO control, read-through drug by itself shows nothing. And we know that why it shows nothing, because this results in full-length protein, but is still misfolded. There is no recovery of function. Interestingly, a combination of modulators like Alexa and Tisa, when that is given in combination with read-through, shown here in this purple line here, you see a remarkable recovery of CFTR function, shown here in this purple line here. And that is why. And why it happens? Because read-through in combination with correctors and potentiators result in production of full-length protein, number one, but that is mature, and that is why there is recovery of function. Next, I'm going to give you a second example where there is nonsense-mediated mRNA decay. The previous example, there was no defect in the mRNA decay. So classic example here is W1282X. This is a pedigree of an, because this is a genetic disease, you can see here this individual is homozygous for this W1282X and parents are carriers. I will try to convey here what is the use and importance of studying carrier parents here in this setting. So we did the RNA sequencing on this individual who was homozygous for W1282X, and the RNA sequencing compared to all the genes that are expressed. You can see here in this red dot, CFTR W1282X undergoes mRNA decay. That is why it falls on the left side of this volcano plot. We had done other simple RT-PCRs that is shown in this publication JCF that came up in 2019. I'm trying to show this as an illustration here that NMD is very complex. There are like around 100 proteins that assemble at this particular PTC, a nonsense variant, and this is a very well orchestration. And this is during the pioneer round of the translation, how this finds that there is a downstream exon junctional complexes, and that results in the mRNA decay. We are very complex. In fact, this is the molecules within us, those are more complex. Giving here, now you, another assay to demonstrate that W1282X undergoes decay, and I was trying to give you a point that what is the advantage of studying the carrier parents, because carrier parents contain one wild type allele as well. So in this chase experiment, after giving a transcriptional shutdown by actinomycin D, what I have observed here in these primary cells is that you see here in the green that this was the transcript level at 0.0. When you chase it for 2 hours, what you see is that the relative expression of the W1282X decreases to almost 0, which indicates that W1282X was undergoing decay. In contrast, shown here again, I'm bringing you back to this illustration when you use an NMD inhibitor that interferes with interactions between these two molecules of the NMD machinery. What we observe is that there is dramatic stabilization of the W1282X transcript. How would that look like on a Western blot? In the interest of time, I'm just trying to show you here, circle, like put in this rectangular box here, that when you add an NMD inhibitor, the truncated protein starts to show up. And when it is used in combination with the corrector molecules, further there is increase in the intensity of these bands. How would we translate this to a clinical situation? So because these compounds, NMD machinery inhibitors could be toxic, so there are antisense oligos that the companies are making, and this is antisense oligo by Ionis Pharmaceuticals. You know that Nasal, that Ionis Pharmaceuticals have developed for SMA is already in use. So they have developed these antisense oligos that can inhibit the nonsense mediated mRNA decay. I have used their inhibitors of NMD. Here I'm showing you a SMUG1, which is an antisense oligo against one of the NMD machineries. Again, with this purple line, what you are seeing here is that the majority of the recovery of CFTR function in W1282X is when you add the combination of NMD inhibitor, a read-through drug G418, and the corrector molecules Alexa and Tisa shown in the purple line here. And that was in the CFBE stable cells. On the right, I'm showing you nasal epithelial cells of these individuals, and there is a recovery about 19% of the CFTR function. So that was all small molecule drugs. And as you know, that there are gene editing approaches as well that are coming up. And I have tried this, one of the editing approaches. And what I'm showing you is that one of those types, and that is adenine deaminase based gene editing. What it does here is that you use a single guide RNA that targets a Cas adenine deaminase fusion protein that targets a particular region of the genome. Adenosine is deaminated to inosine, which is converted to guanine by cellular DNA repair machinery. Again, I'm trying to bring back to our basics. The biochemistry has taught this, and that has been applied now to these tools that is revolutionizing the medicine. And what this adenine base editing then does, it allows for the conversion of A to G edits and T to C edits. And this is more specific for a W1282X. That is why I have given you this example. This is the work that I have done in collaboration with Patrick Harrison from University of Cork in Ireland. And what we are showing here is that when no guide RNA was added, shown here in the blue line, there is no recovery of CFTR function because W1282X is a disease-causing variant. Interestingly, when these W1282X guide RNA with this base editor were used, you see the recovery of the function, and these are 4 different replicates we have seen, and after adding Forsman and IBMX, there is a recovery of CFTR function. How does it look like when you use a wild type individual, normal, healthy? You see a recovery of like around 15 microamp of the CFTR currents. So when you plot it, how much % recovery of CFTR function you got? So that is like about 12 to 15% of CFTR function. Is it like now it is done case for gene editing? Absolutely not, because it works very good in ex vivo conditions where you can electroporate the cells and then you let them differentiate, and the cells grow, multiply, and get differentiated and show the function. However, in a real life scenario, what you need to do is you have to target the specific cell types. And that is where the nanoparticles, I saw Dr. Sadhna also on this. So nanoparticles are making a big way in these CRISPR approaches, where they are trying to target the specific cell types and better delivery of these reagents through the mucus layer. So what you need to target here are the basal cells so that these can further replicate and then differentiate. If you have to do gene replacements, then of course the cell types you have to target could be goblet cells or ionocytes, which are CFTR expressing by single cell RNA-seq. It has been identified now that these are the CFTR rich population of cells. So if you have to do gene replacements with these viral vectors, so these are the cell types you need to target. Last example, very quick, I will give you on a splice variant that we did in collaboration with Sue Slovenhoff at Harvard, where she developed a deep learning approach. And she found one compound. And interestingly, when she was doing this deep learning approach to find out which other sequence features would mimic what they had found as the targets, they found that one of these variants in the CF gene, which we had shown like in human mutation, that it results in aberrant splicing in late 2014. Looking at that paper, she wanted to collaborate with us. And I did this assay. And on this particular RT-PCR, what you can see here is that on the left two here are the wild type expression mini gene that results in a normal splicing. However, when you introduce this variant 31220G2A, when it is untreated, majority of this is misspliced. Interestingly, when you add their splice modulators, and I have shown this in two different replicates, you see the intensity of this band starts to increase, which is a normal splice product. I have also done, but in the interest of time, I'm not giving you those data, that these increased normal transcript also resulted in more protein production observed on Western blot and recovery of CFTR function. And this is published last year in Nature Communication. So what this has led to a hope for individuals with CF. On left is Danny Bessette in 1989 when he was 4 years old. And like now, about 3 decades later, he's holding that journal Science that appeared, had 3 papers on identification of CF gene, and he's leading a healthy life because of those modulators that have come up. And while coming today for work, I was listening to a podcast, and that podcast, it was said that hope is radical. And by radical, it means it is not limited to one. It changes and it continues, and it is available to all. So what about CF in India? Those people do not have access to these drugs. What you guys need to do, and this is what we had emphasized on. I'm coming back to from where I started, that we need to understand the epidemiology and genetics in the perspective of Asia. We have to undertake functional studies to benefit from precision therapies. More recently, your NGS-based carrier screening has emphasized, and this is from Dr. I.C. Verma, that there are higher carrier frequency of CF in Indian population, which suggests, just imagine even if there is 0.4% of the carrier population, given how much is the population of India, how many individuals of CF you could have. And this is the final data set from exome sequencing that has come up and that has prioritized that, again, the same concept, that there are more carriers of CF in Indian population. So in summary, understanding the diversity of CF is critical for accurate diagnosis, prediction of disease progression, and identification of treatments for each individual. Expanding these treatments will require continued development of alternate therapies beyond modulators. Therapeutic approaches can address the underlying causes of all cases of CF. Gene editing technology represents a possible curative approach for treatment of CF in an agnostic way. And lots of thank you for my mentor, Gary Cutting, amazing graduate students, lab technicians, genetic counselor, administrative assistant Patricia Cornwall, Christian Marlowe, who does the nasal brushing, Shivani, who's a research coordinator, all these physician colleagues who run here, the CF Center, and Laurie here in the middle, who is the director for precision medicine for Center of Excellence. Acknowledgement to funding organizations, Gary Cutting, I have already explained, Calvin Cotton, who trained me for primary nasal cells. And animations that I showed you was from CF Foundation. Thank you very much for your time. If you have any questions, feel free to ask.
Thank you very much, Neeraj, for such a brilliant talk, and most of it done with your own hands. Most of the work which you have shown, and that made us understand quite a lot of what advancement is taking place in genetics of cystic fibrosis, development of new therapies, small molecules, pharmacogenomics, and ultimately having to treat, getting to treat these patients with these. And I was just going through these randomized control trials which have come with Trikafta, that these patients are able to retain so much of their lung function because of these medications. So this talk is now open for comments and questions. I can see Dr. Sadhna, Dr. Indu, Dr. Savita. If they want to ask any question, Dr. Pankaj is also there. He's been now working on genetics also of cystic fibrosis. His PhD students are there. So welcome to this session, and please ask your questions.
My heartfelt thanks to Dr. Indu, Dr. Sadhna, Dr. Jyoti, Dr. Savita. I had a tremendous amount of good time with them while I was doing my PhD, and I cannot thank you enough for all the training that you people have given.
Yeah, thank you, Dr. Neeraj. It was really a wonderful talk, and I really very much happy that it's a great advancement you have done in the field of cystic fibrosis, taking it from the Department of Biochemistry and Pediatrics to the John Hopkins. It's a wonderful piece of work you have done. Great appreciation.
Thank you. My pleasure.
Hello, Dr. Neeraj. Yeah,
Hello.
it was really wonderful listening to you, and you look the same as you were here in our department after so many years.
Thank
So,
you.
really nice piece of work. So I just have a small query.
Sure.
When you say that the shortened proteins, when you stabilize those with this corrector molecules, and they get glycosylated and they become functional.
Yeah.
So if we go through the structure of any channel, here the chloride channel, so the structure is quite complex. It has so many domains, and especially the membrane spanning domains, which have arrangement of various amino acids around the channel, like in terms of charged amino acid and so. So how these shortened proteins can become functional into such a complex structure? So have you seen some bioinformatic using some conformation there, and like that?
Yeah, so that's a great question. So for the other audience, I mean, question that Dr. Jodip is asking is that how does this shortened protein works once you give these read-through drugs? Because CFTR is so complex, it has two membrane spanning domains, one regulatory domain, nucleotide binding domain, and terminus, C-terminus. So first answer that I can give you, just to talk about what you asked. Now, for a moment, let us not talk about the use of the read-through drugs. So what our systematic approaches have shown that, there are certain regions where even if you remove part of the CFTR, it gets dysfunctional. It gets functional. And from where the data are coming. So to treat W1282X, what approaches they are taking? They are trying to disrupt the splicing machinery where this W1282X is located. So they just remove one exon 23. And what they have found is that when you remove that exon, it is still functional and it becomes more receptive to these potentiator compounds. That is one example for that. Second example is that when people were trying to develop gene therapies, they always felt that because CFTR is so big, you cannot put them in these vectors because they have a packaging capacity. So they tried to remove a region from the regulatory domain, about 123 amino acids. They call it as delta R. It is functional in that scenario. One example on E831X that I showed you, that there was one amino acid that gets deleted because of that missplicing that is introduced by that is also functional. Coming back to your question about read-through, I think I apologize if I was not able to convey in that really fast explanation of the results. So I want to convey this clearly, that when N-terminus is deleted, and that is a shortened protein, it is not functional. It is dysfunctional. It gets ubiquitinated. But what that gives is that the mRNA is stable. This is how the protein translation machinery has cheated the nonsense mediated mRNA decay. Because a downstream methionine is activated, now a shortened protein is formed. It does not see any further nonsense mutations downstream. So mRNA machinery recognizes it as stable, go ahead, make your protein. How we utilize that stabilization of mRNA decay is that now it becomes a very good target for read-through drug. Now what read-through drug does, it introduces an amino acid at the position L88. So another amino acid is inserted, then it becomes a full-length protein. It no longer remains shortened. Once it becomes full-length protein, the problem that comes up is that it becomes a full-length, but is still a misfolded because it has introduced a missense change. And that was one of the reasons, and I try to convey this in publications, that PTC Therapeutics had come up with a drug atelurin. They were doing it for those nonsense variants where the mRNA was unstable and they were only prescribing read-through. It never went through the clinical trials. So my proposal, my hypothesis here is that the N-terminal nonsense variants where a downstream methionine gets activated are the ideal targets for the read-through drugs. I know it is a little bit of information, but I will be happy to share my paper with you so that it becomes a little bit more understandable. I apologize. I know it is a little tough.
Okay. No, thank you, Neeraj. It becomes quite clear now. Thank you so much.
Sure.
Thank you, Jyoti, for that nice question. Any further questions you can type? There's a question about clinical relevance of career screening. Dr. Pedya has written this question. Functional characterization for Indian scenario and CFTR modulators. Any sponsorship from CF Foundation for study collaboration?
Always my answer is yes, yes, yes. One should do it. We should, or you should consider seriously for setting up the registries, because each time our CFTR2 sits, they put up a map in which they say that, you know, how many countries are participating in that CFTR2. We have now close to 90,000 or even 100,000 individuals worldwide. But India always comes in that shade where they always say they are trying to communicate with them. So career screening, always you should do it. You should talk about this. You should emphasize on this. Even if they say that, you know, there are less patients, you have to talk about that. The data is suggesting there are more carriers. We have seen it. I still remember an individual who had infertility problem, had found Delta F508 in him, had found Delta F508 in others. You should go for it. In terms of sponsorship, I think it is always like, you know, write one paper, communicate with us, or communicate with others, everybody is open to collaborate. And this is how it works. You can talk to Dr. Indu. She's very good at that.
Yes.
Dr. Neeraj, it was wonderful talk. I'm so proud of you.
My pleasure.
You
Again, I'll come back to it. Is all teachings that I have received from PGI.
I have a question. Like you have shown that modulators are really doing impressive work as far as survival of patients is concerned. You showed a wonderful graph where it has been shown that with the introduction of these modulators, survival has really increased in these patients. My question is, this is about mortality data. What about morbidity? Are they still, I mean, what about their susceptibility to opportunistic infections? I mean, CFTR is still fully functional in these patients, or there is, to some extent, their functionality is recovered by use of these modulators. So what about morbidity in these patients?
Yeah, so again, I think this is a very good question. There's a lot of variability in this. And on the outset, I will say that, you know, since I do not see patients, I will not be able to give you a direct answer. But what I have heard from our pulmonologists here or from others at the CF meeting are two things. That, for example, CF-related diabetes, that still remains a problem because pancreas have already got atrophied, or it is already insufficient, so it does not recover that. That is one part. Second is opportunistic organisms. It always remains a chicken and egg situation that, you know, whether it is the dysfunctional CFTR that allows bacteria to come, or it is the bacteria that comes first. So there, it's like PFTs have improved dramatically. But what is even more interesting is that there has been some individuals in which there has been not a recovery, even among Delta F508. They do not respond to these drugs. There is a cohort of those Delta F508. Why they do not respond? Again, like, you know, how this drug gets metabolized in those individuals, what are the other modifiers, what is their profile, like how bad are their lungs. So those are the situations that are still there. It's not like everybody responds. And your question is so very well taken, that groups that are moving forward, even Vertex Pharmaceuticals, though like I have a conflict of interest that I'm funded from Vertex Pharmaceuticals, but all of them are moving really strong with gene editing approaches because they feel like, you know, just treating the basic defect like really is more important than throughout life giving these drugs, because nobody knows after, you know, a follow-up for many number of years how those individuals will come up. But at this time, I think the individuals who are responding to these drugs, I think they benefit a lot. So there's no doubt about it. But there are a handful of people who do not. But again, coming back to your questions, I do not have a very direct answer that has really the microbial colonization decreased or not. I'm sorry for that. Yeah.
Actually, Neeraj, I think that depends on when the drugs are started, because this is a congenital, you know, problem in which things have taken place and things are taking place right, you know, in the infancy.
Yeah.
And hence many of these drugs can't be started right there and then. They are started late. And because of that, they've already had those issues with infections, inflammation, and some deterioration in pancreatic function right in the beginning. So that is why I think they're thinking of gene editing right at the germ level, so that, you know, the individuals born would be free of the defect. So, yeah, that is another approach which is being taken for, you know, for genetic interventions. But as far as the trials are concerned, many of these drugs have really shown very wonderful results, and PFTs have improved, quality of life has improved, life has been prolonged. So obviously, these drugs have a lot of role to play. And I think we should try to find some drugs. In India, we have a very big traditional system, which had many drugs, and people did talk of number of drugs of those kind. But these drugs, when they're going to reach India and are going to be within the financial reach of our population, is something which is, but we are taking it up with the government that they should try to fund some of these rare diseases treatment as they are now doing for SMD. So I hope cystic fibrosis also comes in that category, and they can get these, our patients also can get these drugs. So there is a question by Gursimran Kaur, for nonsense variants, should we have to start with read-through drugs only and then go for NMD inhibitors?
So I will go, again, a good question, and thank you for asking. I will go the other way around. NMD inhibition is first and then the read-through. Again, for those variants where there is an evidence that they will undergo decay. So any nonsense variant that undergoes decay will have to have NMD inhibition. But this comes with a caveat that, NMD inhibitors are kind of, NMD inhibition is at the endogenous level is also very important to control the quality of the transcriptome. So please, 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 I'm not saying that tomorrow start giving NMD inhibitor to an individual. There are alternate pathways that need to be identified so that it is not toxic to people. NMD, is very important for the normal transcriptome of individuals as well. But answer to your question is that if you find that any nonsense mutation that undergoes decay, so a textbook answer will be use NMD inhibitor and then add read-through. Only then read-through will work. But thank you for asking.
Thank you very much. Neeraj, I think it must be a little late for you now because what's the time right now for you? I can see a coffee machine at the back. So
it's midnight. It's midnight, 12.
Oh, okay. So, thank you so much for being with us at the late hour. We know you are quite used to working overnight, and it's actually this midnight oil, I guess, which has paid you, and your devotion to work. And I think
sure, always. Thank you very much.
And we wish you the same, the same speed and the same heights for you in future. Thank you very much for joining us. Dr. Balram, would you like to say something?
No, ma'am. Just I opened my video actually. That is all.
Okay, thank you. Thank you very much. Thank you very much for being here. Thank you, Neeraj. Thank you so much.
Thank you. Thank you.
Good night. Yeah.
Have a good one. Bye.